So enzymes are an important group of proteins. Not all enzymes are made out of protein, but almost all of them are. In some of my previous videos, I've talked about the process of metabolism. Metabolism is really just the sum of all the chemical reactions that occur inside of a cell. Enzymes are absolutely necessary to speed up these chemical reactions. The chemical reactions that are needed to sustain life do not occur fast enough to actually sustain life without the presence of enzymes. So all of these chemical reactions that occur in our body, they do occur naturally, but without enzymes, they would happen so slowly that we could not exist. So enzymes are really important in biology. We say that enzymes increase the rate of a chemical reaction, and we call this catalysis. And as I mentioned, that in order for any chemical reaction to occur at a rate compatible with life, an enzyme is required. If you kind of think of it this way, and a chemical reaction without an enzyme is sort of like using dial-up internet. It will work, but it will take forever, and it will make things very ineffective, and why did you even bother doing it, right? Whereas a chemical reaction with an enzyme is like using a broadband internet connection. It's fast, it's speedy, it's efficient, and it allows you to get stuff done, right? The way an enzyme works is it kind of acts as a stage on which a chemical reaction will occur. Right? I don't know if you've heard of the saying, if you build it, they will come. That's kind of the idea here. An enzyme is a stage that's been built for a chemical reaction to take place on. So as long as we have that stage, our reactants will come to that stage and participate in a chemical reaction. So they're basically just providing a location for a chemical reaction to occur. So the reactants have a special name in an enzyme catalyzed reaction. They're referred to as substrates. So using my stage analogy, you can think of these substrates as being the actors in the play. The stage itself is the enzyme, and the play is the reaction. Now, in this particular play, the actors have to stand in one very, very, very specific place on the stage. And that's the only place they can stand, or else the play doesn't work out. In an enzyme, we call this an active site. So the active site is the spot on the enzyme where the substrate will fit in. Substrates are very, very, very specific to the active site on their enzyme. And it's much like a lock is very specific to a key, right? The key is like our substrate here. It has a very specific shape. And the lock is our enzyme. It also has a very specific shape that will only fit that one particular key. So an enzyme usually only fits one specific set of substrates. Once the substrates are sitting in the perfect spots on the stage, basically they're almost touching at this point, and that's what allows the chemical reaction to speed up, because we've brought these reactants, these substrates, really close together, and now they can simply react with one another and they form a new product, which is then released by the enzyme. Enzymes have a lot of really cool properties. They can catalyze both anabolic and catabolic reactions. So remember, catabolic reactions break things down and anabolic reactions build things up. So an enzyme can be used to do both. Actually, one enzyme can be used to either break down or build up the same exact reaction. It just kind of depends upon the conditions in the cell, which direction it's going to go. As I mentioned before, enzymes are very, very specific to their substrates. Enzymes are also very sensitive to temperature and pH. They are made out of proteins, and as I mentioned before, proteins can become denatured or unfolded when there are changes in temperature and pH. So each enzyme has a very specific temperature and pH that it works best at, and we call these the optimal temperature and the optimal pH. If the pH or temperature is anything but optimal, the enzyme will work less. Enzymes can also be regulated. So for example, there are substances called inhibitors, which when they bind to the enzyme, they prevent the enzyme from working. So we have a lot of different ways that we can regulate enzyme activity. We can turn enzymes on, we can turn them off, do a lot of different things with them. It's a good way to kind of regulate metabolism is by regulating enzymes. I think one of the coolest things about enzymes is that they can be used again and again and again and again and again, right? So a lot of students have this misconception that once an enzyme is used in a chemical reaction, that it falls apart and never to be used again, but that's not true. Just like a stage can be used again and again and again, this enzyme can be used again and again and again. And one important thing to emphasize is that enzymes don't actually supply any energy to chemical reactions. Again, this is another misconception that students have. Enzymes speed up a chemical reaction by bringing substrates together in such a way that they're more likely to react, but they're not actually providing any energy to that reaction. There's no ATP, for example, coming into that reaction to make it happen faster.